With chapter 15, we're going to be stepping back from examining closely bacteria, viruses, the different parasites and whatnot. We're going to be looking at, well, microbiology from the medicine standpoint, the medical community standpoint, the administration of how we go about looking at and examining things. Now, we need to keep in mind that knowing what the infection is, what's causing the infection, which is thus causing a disease, that is the most important part of treatment, of performing medicine. Okay? You can just give anybody pills, pill, let's cut you open, let's do something to you. But unless you know exactly what it is, why you're doing something, well, that's going to be a bit of an issue. Now, when you go and you look at the list here of 3 methods, we're going to examine the first part of this chapter on how to identify the bacteria. Okay? We have to know what the bacteria is so we can prescribe the correct antibiotic. Okay? If you don't have the correct antibiotic, you could be prescribing the absolutely wrong thing. Because remember the whole narrow spectrum, broad spectrum. If you choose a narrow spectrum antibiotic and it's the wrong spectrum, you give an antibiotic that's great for treating gram-negatives, but it's actually a gram-positive infection, it's going to have absolutely no effect. So we need to know exactly what's going on. And to do that, as you see here, we have 3 ways. We're going to look at the phenotypic. Okay, somebody is going to take a sample, send it off to what's referred to as the ID lab in most hospitals, the infectious diseases lab. A tech in the infectious diseases lab is going to make a glass slide, kind of like what you've been doing in the virtual labs, or what you have done. Make a slide and then gram stain it. Okay? Going to look at the gram stain, going to try to determine what it is. Probably. Kind of get a good idea. But we're not going to know exactly what it is just by looking at the gram stain. We'll be able to tell, is it cocci, bacilli, coliform, is it one of the alternate shapes, Is it a single cell, or is it one of those that clusters, or is it in chains, things like that. Gram-positive, gram-negative, okay? Start off with the gram-positive, gram-negative, okay? That splits a lot of what could be our infection. Okay, whether it's a skin infection, an abscess in the mouth, maybe they have cardiac infection, whatever. Endocarditis, I believe. Yeah, endocarditis. We need to know gram-positive, gram-negative. Okay. Then you're going to look at the shapes. Based on the shapes, it's a gram-positive, but it's a bacillus. Okay, that really limits, gram-positive bacilli, gram-positive rods. There's very few of those that can infect humans. And our list of possibilities gets shorter and shorter. Then we're going to use maybe immunological, okay, antibody testing. Turns out, as we're going to see in a later lecture, antibodies are highly specific. They can be highly specific for certain types of bacteria, or they can be species specific. Serotype, different versions of different species, subspecies specific. And within the past few decades, we've been getting more into the genotype. What's the DNA say? What are the specific sequences in the DNA? And that's what we're going to be talking about in this chapter.